All right, so this is going to be our last book lecture of the semester, and this material kind of jumps around, and I talk about a bunch of different topics. Some of them I feel like that are kind of important for you to know coming out of a basic biology class, since they're kind of relevant to what's going on in today's society with science. So we are going to kind of jump around here. So you can see what sections if you want to read further, because maybe you don't understand what I'm trying to say here with the heading here. So this would be chapter 11, section 2. It talks about modifications that that affect gene expression. So we talk about X chromosome inactivation. So in female mammals, one of the two X chromosomes is highly compacted and transcriptionally inactive. What does that mean? That means females have two X chromosomes. We know if you have too many chromosomes, it causes problems. For example, Down syndrome. We know that you can live perfectly fine with one X chromosome because males do. So when females have two X chromosomes, that is too much information. So what happens is one of those X chromosomes get turned off. So they wind up and don't get expressed. So none of the information on there can be read to be transcribed into messenger RNA to make additional proteins. Now, this decision on which X chromosome to turn off happens very early in embryonic development, but it's not all moms, so not all cells have moms on and not all cells have dads on. It is a mixture. It's a 50-50. So if let's say we had 1,000 cells at that part of the embryo and they get the signal to shut one down, you could have a 50-50 if 500 cells have moms, 500 cells have dads, or whatever percentage, okay? Just one of those X's get broken down. And so here we have our structure of DNA, our unwound DNA. They wrap around these histones and then they form that chromosome. So the DNA in the inactivated chromosome winds all up and stays wound up. Now, this X inactivation, we can also alter, well, we'll get into the other stuff here in a minute, but again, we're going to wind up around those histones. And so notice here, I'm nice and wound up. I can't transcribe. I cannot make a copy of messenger RNA. Versus here, we're nice and we're open and we can then transcribe those genes. So that's the difference between being open for business and making protein synthesis, undergoing protein synthesis, or closed and not able to make those proteins. So we refer to this as dosage compensation because females, again, have two X chromosomes, so we have to turn one off. Males only have one X chromosome, and of course they got that from their mother, so they have that female's X chromosome. So in a female, or we can take cells and we can look at these things called Barr bodies. And so Barr bodies are the inactivated X chromosome. Not only are they wound up, but they also kind of attach themselves to the nuclear membrane. And so we can do a test to test for whether you are genetically male or female by what's called a Barr body test. Males will not have that Barr body, females will. And they actually did use this test back, I think in like the 60s during the Olympics, if I got my dates right. They wanted to make sure that there were not males pretending to be females so that they would be at an advantage in the games, the Olympic games. So this is referred to as a Barr body test. We also see this phenomena in kitty cats. So with our kitty cats, we have this thing called either a calico cat or tortoise shell cat. And with those cats, those are all going to be females, because if we look at the colors, whether you're going to be orange or black, males can only be one or the other, because notice here, this is sex linked. So the gene for fur color is on the X chromosome. So here, again, a male only has one X, and in this case, he has the black copy for fur color. In this case, he has the orange. Over here, if I could be homozygous for black, I could be homozygous for an orange. But what happens if a female is heterozygous? So remember, half or approximately half will have dad's X chromosome and half will have mom's X chromosome. So we will get splotches, and you can see, like, for instance, maybe all the orange splotches are the cells that kept mom's X chromosome, and the black cells would be all the ones that kept dad's X chromosome. So that is how you get a calico or a tortoise cat. So here's a little tortie here. So we can see that we have some patches that kept the black fur activated, and some that kept the orange. Oh, yeah. Keep yawning here. This weather, it's another rainy day in Illinois. We all just want to curl up in bed and watch TV all day long. All right, so anhydrotic dysplasia. So a lot of people will ask me, well, do we see something like this in humans? And we do. We do with this thing called anhydrotic dysplasia. And this means they do not have sweat glands. So if you have a normal X, you have normal sweat glands. If you have X prime, you have the absence of sweat glands. And so you either have a male, again, either has it or they don't, because they cannot be carriers, right? They have only one X chromosome, but females could be heterozygous. And if they're heterozygous, that means they are what we call patchy sweaters. They have patches on their skin that sweat and other patches that do not. And then we're going to kind of leave that whole thing with the X chromosome, which is a little bit more of our genetics. We're going to get into genetic engineering and cloning. And we hear about cloning and recombinant DNA and stuff all the time. So I kind of want to give you an idea of what that means. So I need to do some jumping jacks or something here. So, you know, when you guys think of cloning, most of you guys think of whole organism cloning. Of course, cloning humans is illegal, but we do have the ability to clone cats and dogs and other organisms today, which is very expensive, and it is much cheaper just to make a cat or a dog the old-fashioned way. But we can, in fact, do some mammalian cloning. What is actually more frequent that we do is we clone just genes. And so I'm going to kind of, I like pictures so much better. So in these pictures I'm going to show you, I will explain how we do this. All right, so this is the steps, not using the pictures, but first we're going to take the plasmid. Now, a plasmid is a small circular loop of DNA we find in bacterial cells. Plasmids often carry miscellaneous genes. Not any genes they need to survive, but genes that, or I should say housekeeping genes, but they may have genes in there, and many of them have genes for antibiotic resistance or toxins, or they're nasty little things in those plasmids. So first of all, what we're going to do is we're going to get a DNA plasmid. We're going to locate the gene that we want to clone. We're going to use these things called restriction enzymes, and restriction enzymes are going to cut the gene we want out of the DNA, and we're going to then put it inside the plasmid. And then that is called recombinant DNA, because we now have bacterial genes mixed in with a different species of genes. We are then going to use the bacteria to make more plasmids, so they are going to reproduce that, and those are clones. All right, so we're going to have the bacterial cell do the cloning for us. So here's a picture. So here is our bacterial cell. So this is E. coli. This is the normal genes that the cell needs to survive, and this is extra little loop of DNA we find that often has some extra information, like I said, antibiotic resistance or toxins. Here we have a human gene, which we'd have all 46 chromosomes in there, and we'd have to locate the gene that we want. And usually that's half the battle, is finding in our genome where that gene is. and isolating it. So here we have our plasmid isolated. Here we found our gene in our human cell. We cut it out. We inserted it in the plasmid. We then put the bacteria through some procedures that literally punch holes into the membrane, and then we use temperature differences to cause kind of like a vacuum, and it pulls that plasmid in, and then we let the bacterial cell recover, and then it starts to replicate, and as it replicates, it's making more of those plasmids, and now we have those genes, and we can do several things with those genes. So for one, we could take those genes and cut them back out and put them in another organism, or those genes may be used to actually make that protein, and so we harvest the proteins that the bacteria are making for us. So here is a bigger picture of that. Here's our plasmid. Here's the gene of interest that we want right here. We're going to cut them out using restriction enzymes. Restriction enzymes basically find sequences in DNA and cut it. We will insert it inside the plasmid, and then we're going to use DNA ligase, which we saw that when we attach our Okazaki fragments together when we replicate DNA. So we can use that to recombine this DNA. And now we have our recombinant DNA plasmid. Once we have that, we get it taken in to a bacterial cell. We call it a transformation. We kind of talked about transformations when we went from rough to smooth in our Griffiths experiment. And then our bacterial cells replicate themselves and that plasmid. We then again can take out the replicated gene, or we can have it actually make those proteins we're interested in having made. So recombinant cells in organisms constructed by DNA technologies are used to manufacture many useful products. Mostly what we're trying to make are proteins. Bacteria are often the best organisms for manufacturing a protein because of the plasmids they contain. And we can also use phages. They infect bacterial cells, so we can use the phages as a way of delivering those cloning vectors that we want. So it's very cheap to grow bacteria, and nobody's really the human rights activists, right, aren't gonna have a problem with us growing and killing bacterial cells. So it's very convenient, and then often the bacterial cells will literally just secrete the protein that we're interested in into the growth medium, because we tend to grow them in like this broth. So they basically kick that out into the broth. Now we can also use, so far we've been talking about using prokaryotic cells to do our cloning. Well, sometimes that's not possible because we talked about introns and exons and stuff like that. And so a lot of times that messenger RNA has to be processed to cut those interrupting introns out, and prokaryotic cells do not have ability to do that because they do not have introns. So we would need a eukaryotic cell to do so. So we're going to use yeast cells, and yeast cells can often then get around that problem of that processing that messenger RNA correctly. Pharmaceutical researchers are currently exploring the mass production of gene products by either, like I said, whole animals or plants. We also have what are called recombinant animals. Again, they are difficult to make and very expensive. So here we do have a picture of some cloned animals. And so with that cloned animal, so our first cloned animal was Dolly, and you guys may or may not have heard of Dolly. That was our first mammal that we cloned. And what we did was we took an egg out of our white-faced sheep. That stupid link keeps showing up. I'm trying to get it away. So if we look at A, basically we harvested an egg. Okay. Actually, A is the it's not real clear here in the picture, but A is what they did was they took a diploid cell, a regular cell from her mammary glands, actually. And then they took an egg from the black-faced sheep. Now, black-faced sheep are dominant. So if they took a purebred black-faced sheep and bred it with a white-faced sheep, you'd always get black-faced because, right, it's homozygous dominant. So all her offspring would have black faces. So they took her egg, they took the nucleus. So notice B shows you an egg with no nucleus. They took the nucleus out, and then they inserted the nucleus from the white-faced sheep. Then they got that cell to undergo mitosis, and it developed into a whole sheep fetus, which they actually took those cells, those developing cells, and they put it back into the black-faced sheep's uterus for it to harvest that baby. So incubated Dolly, and then she gave birth to that clone. That, if you notice, the baby has a white face on it, that is an identical clone to the original white face adult sheep. So that was Dolly. We actually learned a lot of stuff about Dolly, but that had nothing to do with cloning. But we got a lot of interesting information about things called telomeres and stuff like that. But like I said, it's very expensive to make these cloned animals. So even though you could go online and you can pay, I want to say it's like, my son looked into it. I think it was like maybe 16,000 if you want to clone your cat, and I think it was 20 something for a dog. So very, very expensive to do. So this is just a chart that shows you some of the things that we use recombinant. We use this recombinant DNA tech to do. So most of you guys know about human insulin. We used to get insulin from cows. It was cow insulin, and some people reacted negatively to the cow insulin. So now we actually found the gene for human insulin. We make it in E. coli, and now we have a very inexpensive way to treat diabetes, although we hear a lot about the markup on this product. We also have human growth hormone, and so we use that to treat growth defects. We have our epidermal growth factor, which is used to treat burns and ulcers to speed up growth from that type of cells. We have cancer treatments. We have Taxol, you guys may have, is a real famous cancer treatment for ovarian cancer. And then interferons, another thing we use for cancer, so a lot of cancer here. Erythropoietin is a treatment for anemia. And then factor 8 is for hemophilia. We talked about hemophilia in our genetics chapter. And then you guys have heard of TPA, tissue plasmagen activator, that is used for heart attacks and strokes. So we also are going to make this product using this technology. So mammalian cells must be used, and we saw that at the bottom of the chart that had mammalian cells, because in mammalian cells, we may have to do some extra doctoring. If you remember when we talked about protein synthesis, we have some proteins that will stay into the cell, and some that go into the membranes or the organelle membranes, or inside an organelle. They go to the rough endoplasmic reticulum. They then go to the Golgi apparatus. And so we still need to have that pathway for them. Many times they have to add sugar chains or other modifications to that protein to make them active. And so that's where we would use these mammalian cells. And then here in your book, it talks about spider goat. So this particular goat is a recombinant organism. We know that spider webs, the silk, is one of the strongest materials on our planet. But you cannot really have what we call a spider farm and harvest spider webbing. For one, most spiders are carnivorous. They eat each other, cannibals, I should say. So you'd have to have one spider per tank, and then, yeah, it just wouldn't be cost effective. So they found the gene for making spider silk, and they inserted it into this goat, and this goat literally produces that silk in its milk. And then they will strain the milk to get the silk out of it. So kind of creepy. So again, therapeutic hormones produced by DNA technology. I talked about insulin to treat diabetes and HGH, human growth hormone, to treat dwarfism. It helps get them a few more inches of their height. We are also using DNA technologies to test for inherited diseases, to detect infectious agents, and we are working on producing vaccines or other derivatives of a pathogen to stimulate the immune system to be able to fight certain infections. So lots of things that we're using all this molecular biology to do now, which is why I kind of wanted to make sure we talked about this. So we get to these things called GMOs, genetically modified organisms. GMOs are organisms that contain one or more genes artificially. Now, another word you may hear is transgenic or transgenic organisms. And these are organisms that contain at least one gene from another species. Okay. So in this case, we have these mice and we inserted some kind of luciferase or other glowing type of protein from a different type of organism into the mice. So one of the most common vectors used to introduce new genes into plants is a soil bacterium called Agrobacterium tumefaciens. And there is a plasmid called the TI plasmid inside it. And so here we have our Agrobacterium. We get that TI plasmid out of it. We're going to do the same thing. We're going to use a restriction enzyme, cut it open. So we open the ring. We're going to use that same restriction enzyme to cut out our desired gene. We're going to use our DNA ligase to put it together. And then what we're going to do is we're going to introduce that recombinant DNA into our plant cell. And once we deliver it to the nucleus of that plant cell, we have the new DNA inside it. The cell will then grow into a plant. And of course, every time it replicates, it will or undergo mitosis. It's making a copy of itself. And so now we have a plant with that new trait. So we have what are called GMO plants, genetically modified organisms. What we can do is we can put resistance to herbicides and pesticides inside our plant so that we basically what they're doing is they are making them resistant to Roundup. I'm just gonna call it what it is. We're gonna make it. We have what's called BT corn and some other types of products. And we have put these genes inside them that make them resistant to Roundup. So now we can spray Roundup on the fields. It kills all the weeds. It keeps the corn, soybean, and other organisms alive. The problem is people do not know how, well, for one, you're spraying high amounts of herbicides on these plants. For two, we are not sure how those newly introduced proteins and genes inside that plant will affect humans. So this is where people are getting all scared or panicked or cautious about eating GMO foods. Okay. We also have the same thing going on with animals where we have put in inserted genes that you don't normally find in those animals into them. And so again, how does your digestive system handle that new type of protein? So that's a big question that nutritionists and other people are arguing back and forth about. We know some people can eat genetically modified stuff and be perfectly fine. Some people cannot. Some people, we may not even know like what it causes until 20, 30, 40 years down the road. So a lot of people err on the side of caution and try to stay away from GMOs. This is why food products today have to put on their products that they are a genetically modified organism. So here, introduction of allergens into the food supply and the spread of genes to closely related organisms, because what we're finding now is that if you have one field that has genetically modified corn and you are a neighboring farmer and you have organic corn, we know we can't keep the pollen contained when it's that time of the year when they're pollinating. You cannot keep the GMO pollen to stay put in their field, and so you get cross-pollination and you get contamination of GMOs into what should have been an organic corn farm. So they don't have a way to control this spread in certain cases. So there's a lot of regulatory agencies trying to address the problem, the safety, the labeling, and the safe use of this biotech that we have now. We also have a thing called gene therapy. And so we've been talking about a lot of genetic disorders where the protein isn't made properly. But what if we could insert into that cell the good copy of the gene? Wouldn't that be awesome? And so this is something that we're working on when we talk about gene therapy. We're supplying that functioning allele, that functional copy of the gene into the cell. So what we're trying to do is, viruses are constantly entering your body and inserting their DNA or RNA into your cell, and then your cell makes the viral particles. So, if we could use a virus as our delivery system, take out the bad stuff of the virus and then use it to deliver the genes we want it to deliver, which is the functioning allele, this would be ideal. So we have tried this in a certain number of cases. So I think okay. We're going to take the normal allele, take it and put it into what's called a retrovirus. Retroviruses deliver RNA. And so when we put that virus into our cell, we also have to put another protein in there called reverse transcriptase. And that does exactly what it sounds like, right? Just transcribing goes from DNA to RNA. If we do reverse transcriptase, we're going to take RNA and make it into DNA. And so then that gets inserted into the gene, into the chromosome here. We talked about that lytic and lysogenic life cycles. So remember, the lysogenic puts that copy into the host cell's DNA. So it is going to put it into the cell's DNA, and then we're going to take those cells and inject it into the bone marrow. So like a lot of times what they'll do for leukemia is they will undergo radiation, kill all of the bad bone marrow, and then they're going to inject in here our cloned, our genetically modified cells that will repopulate, hopefully, all those bone marrow cells with the healthy bone marrow cells. So that is what we are attempting to do. So our first successful gene therapy, so it's been 20 years. So you can imagine we haven't really cured genetic disorders, right? So it's still something in its infancy. So they tried to treat 10 children with what's called the severe combined immune deficiency. These are those kids that have to live in the bubbles where they have no immune system. It did help 9 of the patients, but it caused leukemia in 3 of them, and then 1 of them did die. The problem we have is we can't control where that gene gets inserted into the cell's DNA. If it gets inserted into a gene that your cell needs, then it will not work properly. And in this case, it resulted in death. So, like I said, we still have a long way to go in this particular area of science. So then we also have other questions, and we start getting into the ethic part. So how can we build in gene control mechanisms? So again, not only are we needing to insert the gene, but we didn't really talk about a lot of the control, like when does that gene get expressed? How much do we express that gene? Those things come in the DNA sequence just before the gene. Well, we really didn't talk about promoters and operators and all that good stuff, but we have to be able to put those control mechanisms as well as that gene. And then the second one is what I already talked about. How can gene insertion be performed without harming other cell functions? We can't control exactly where it gets placed into the DNA. Will gene therapy lead to efforts to control genetic makeup of the human population? So now we're tweaking our human genomes. Should we try to eliminate genetic defects in our children and descendants when genetic variety is a necessary ingredient for the survival of species? So we talked about sickle cell. Sickle cell was a mutation that does not allow the red blood cell to work properly, but it also allows individuals that are heterozygous for sickle cell to survive malarial infections. So not all mutations or disorders may be bad. Something may come out, and that particular disorder, that group of individuals may be very successful because of that disorder or that mutation. So should we try to eliminate them? So lots of different questions we have. So again, if that is your child that has it, yes, you would want them to be healthy. But, again, when we talk about evolution of the species, we're not talking about an individual, we're talking about the population. And then that brings us to our evolution stuff that we already did. So if you have any questions, again, send me an email so that I can address them. But this is pretty much it for the semester. I do hope you had learned a lot in this class, and good luck on your final exam.